Module 4, Part 1. In this module, we are going to discuss a cytoskeleton that gives the cell its shape, motility, aid in cell division, cell growth, cell interactions, and we will also learn more details of how the secretory vesicles full of proteins are transported with the aid of cytoskeletal components to complete our understanding of the vesicle transport from the last module. You will be able to appreciate the numerous functions performed by the cytoskeletons and why they are so important in this module. The ability of the cells to adopt a variety of shapes to move in certain directions according to certain signals, to carry out coordinated directed movements, to divide by binary fission, and organize their intracellular space all rely on a cellular structure known as the cytoskeleton. The cytoskeleton is made up of a number of proteins that polymerize into filaments. So the cytoskeleton is filamentous. The word cytoskeleton invokes the image of a very rigid structure like the skeleton of an animal. But this nomenclature is unfortunate because in the case of cytoskeleton, it is not a rigid, unchanging structure, but it is a very dynamic and complex network of proteins and filaments. They extend throughout the cell. They shrink and grow as the cell needs it for whatever cellular function is being carried out. So please keep in mind that when you are studying this topic, the cytoskeleton is not a permanent, unchanging structure in the cell, but it is a very dynamic and continuously changing according to the needs of the cell. So this is an amazing fluorescence micrograph of a cell. The cytoskeletons are stained with a fluorophore, probably attached with an antibody, the antibody is attached with a fluorophore. You can see a dense network of filament found in the cell, and they can take different shapes and organizations. And in fact, there are 3 types of filaments that make up the cytoskeletons. So this is an overview of the cytoskeletons of an epithelial cells. And in the top panel, you can see epithelial cell, and you can see a migrating cells at the bottom. In the illustration, these microfilaments are in red, so right here, and you can see that the microtubules are in green. And the intermediate filaments are right here. They are in dark blue, and the nucleus is the light blue oval. So the distribution and organization of the cytoskeletal filaments structurally polarizes different types of cell. For an example, for an epithelial cells lining the intestine have a polarized morphology that we already know, and junctions with all neighboring cells. This morphology and arrangement provide a tight single pavement-like epithelium. Nutrients such as glucose are important from the intestinal lumen across the apical plasma membrane, exported across the basolateral membrane into the bloodstream. So the highly polarized intestinal cell structure with distinct apical and basolateral domains is supported by the cytoskeleton. Core bundles of actin microfilaments provide the support of the intestinal apical cell microvilli right here. You can see that on the transmission electron micrograph on the top right here. These are the microvilli and the actins or the microfilaments that you can see. At the bottom is a migrating macrophage, which is one type of white blood cells moved into the tissues. And we have talked about this phagocytic cell a lot of time. And they do have a complex array of actin microfilaments at the leading edge for the process of chemotaxis and phagocytosis to pursue, capture, and destroy infectious agent or old cells. The TM shows a leading edge of a cytoskeleton as visualized by extracting the membrane and soluble components from the cell by detergent treatment and then making a metal replica of the remaining filament. Its morphologically distinct domain in a migrating cell polarizes the cell structure with a leading edge oriented towards the direction of the locomotion. So here you can appreciate with the chapter opener illustration, this is a low magnification immunofluorescence micrograph showing the section of a mouse intestine stained for the actin in red, the extracellular matrix protein laminin in green, and these DNAs in the cell in blue. So do not get confused that these finger-like projections are the internal wall of the intestine. The whole thing is the internal wall of the intestine. This is the outer wall of the intestine lined by the smooth muscle, and the smooth muscle also do contain actin or the microfilament. We're going to talk about that name later. The microfilament, which is a stained red. On the other hand, here, these are the apical border of the cells, which does contain the microvilli and stained red. And those cells are columnar cells which have these blue nucleus. And right here, this is the extracellular space connective tissue which contain the laminin. So these are the ones which is lining the intestine, and this is the intestinal lumen. That's the outside of the intestine. So the first filament that we are going to talk about are actin filaments, and sometimes these are referred to as microfilaments. These are polymers of actin monomers. When the monomers come together, they form a helical twisted structure. They are very flexible and dynamic, and they can lengthen and shorten. They are about 7 to 9 nanometers in diameter and visible on the electron microscope. And on the left is a molecular model of the actin filament. And as you can see, that there are 3 individual monomers that has been shown right here. And these are a twisted chain, helical chain of the actin. So depending upon the cell type, you will find the actin filaments or microfilaments in different places in the cell. Say, for an example, for these top 3 panels, these are the polarized cell. So for example, here in these polarized cell, we have been talking throughout the course. You can see the dense accumulation of the actin in the microvilli, in the cell cortex, or in around the edge of the cell. This structure is known as the adhesion belt. So in this case, the actin, being the cell cortex, protects the cell and contributes to the structural integrity of the epithelial tissue that is being made up by these cells. In the case of adhesion belts, they also provide the structural integrity to the epithelial, but adhesion belts have very interesting function during development. The epithelium changes its structure and moves to form tubules during the development, and adhesion belts plays a role which is really important to develop that structure. When we talk about more of a cellular interactions, I'll elaborate more on these adhesion belts. In case of motile cells like white blood cells, right here, you will see an actin in these like in filopodia or lamellipodia. These are the structures that are found on the leading edge of the motile cells, and you can see a thin layer of actin around the cell as well, which provides the structural integrity of the motile cell. In a number of cells, both motile and non-motile cells, actin filaments make up these stress fibers, and they assist in the cell for the adhesion. And again, we are going to talk about these stress fibers more into cellular interactions when we study the cellular interactions. Finally, in dividing cells, you can see the contractile ring is rich in actin filaments. Here is an EM micrograph of a fluorescence micrograph showing the actin filaments within the cell. So this is a fluorescence micrograph, and this is an electron micrograph. And you can easily see and appreciate the actin filaments in each of these structures. And here you can see some stress fiber, and this is the leading edge, which is the filopodia. And here you have this microvilli and a thick network of actin towards the bottom, dense network. The second type of filament are known as intermediate filament, and they are so named because they are of the intermediate size to all 3 filaments. So they're right in the middle in terms of size. They're about 10 nanometers in diameter. They are made up of polymers of intermediate proteins. There are a number of different intermediate filament proteins that we'll elaborate later. But when these subunits come together, they form these rope-like structures, which in the cytoplasm provides a structural support to help them maintain the shape. You can find intermediate filament proteins at tight junctions between the epithelial cells, so they help with cell-to-cell interaction as well. You can also find a layer of intermediate filament and the membrane proteins surrounding the nucleus, and this is known as the nuclear lamina. It provides the support and protects the nucleus from the shear forces that are going on within the cells, such as vesicle trafficking during cell division, etc. The third type of filament that you find inside the cell are the microtubules. They are polymers of tubulin proteins. They are the largest of the three filaments, and they are about 25 nanometers in diameter. They are hollow cylindrical in shape, and they are extremely rigid. They are the most rigid out of the three filaments that make up the cytoskeleton. Normally, when you are looking at microtubules in the cell, they usually appear as very long, stiff filament structure. They usually emanate from a single point in the cell, and the point is in the cell is known as the microtubule organization center, or shortly known as MTOC. A good example of microtubule organizing center is the centrosome. As we talk more about microtubules, I will elaborate on what types of proteins make up the microtubule organizing center. I will elaborate on what types of proteins make up the microtubule organizing center and the importance of the structure as well. So each filament type can be labeled with a specific fluorescent antibodies that are visible with a fluorescence microscope or immunofluorescence microscopy. All three of these cytoskeletons are stained in the same cell with different antibodies and attached with a different colored fluorophore. So what you see that here you have the microfilaments in red that are stained with an antibody specific for actin. Then you have the microtubules that are stained with an antibody specific for tubulin. The third panel shows the intermediate filament that are stained for vimentin, which is an intermediate protein. One thing you might notice is that the pattern of the intermediate filament and the microtubules are to some extent similar, not in distribution but pattern. But on the other hand, the microfilament is very different, and this is true in many cells. The microtubules and the intermediate filaments often display a similar pattern on fluorescence micrograph, whereas the microfilament have a more dynamic structure and changing structure that don't match the pattern of the other two filaments. So next, what we want to do is to take a look at each type of filament and elaborate on them. So in this part, we are going to talk and elaborate about the microfilaments. I mentioned that these are helical polymers of actin, so a single type of polymer makes up of actin filament. The monomer is often referred to as globular actin or G-actin. When it is polymerized into a filament, it is known as a filamentous actin or F-actin. So if you hear me say G-actin, you know I'm talking about the monomers. But if you hear me say F-actin, you know I am talking about the polymerized filament like shown here in the picture. So the two ends of the actin filament are not biochemically equivalent. So one end is known as the plus end and sometimes referred to as the barbed end. It is known as the fast-growing end, especially in vivo when the filaments are growing. They are growing very quickly at this plus end, so monomers are being added at the plus end. On the other end is known as the minus end, and sometimes it is referred to as the pointed end. It grows more slowly, both in vivo and in vitro, than the plus end. In fact, for all intents and purposes, in vivo or inside the cell, the monomers are usually not added at the minus end. So how is it that these two ends are biochemically different, and how is that important to their function in the cell? So to understand the difference between the two ends, we have to first look at actin polymerization in general. So actin polymerization takes place in 3 steps. The first step is known as the nucleation. So what happens in the first step, and this is the rate limiting step, or this is the slowest step, you have a group of G-actin monomers and a few of them will come together to form this trimer polymer. Sometimes it is also known as a seed or a nucleus, and this step is known as nucleation. Once this happens, polymerization will occur much more rapidly. So then in vitro in a test tube, what you will observe after nucleation is the second step, the elongation phase of polymerization, where monomers are added both to the plus end and the minus end, and you get rapid growth of the filament. Again, let me stress that in vitro you can observe the addition of monomers at the minus end, but this is rarely observed in vivo or within the cells. So you have the addition of monomers right here. You have the addition of monomers, and eventually some monomers will start to come off, and you will reach a third stage of polymerization, which is known as the steady state. The steady state is when the rate of monomer being added to the filament equals to the rate of monomer that you are losing from the filaments. So you don't get any growth, or you do not get any net growth in the length of the filament, and this is known as the steady state. Now, steady state happens at a certain G-actin concentration, and this G-actin concentration is known as the critical concentration. It is the concentration of monomers at steady state and known as the critical concentration. Again, repeating, it is the concentration of the G-actin monomers at steady state that is known as the critical concentration. So you can think of these three steps in active polymerization as a biochemical reaction. Shown here, G-actin being converted to F-actin and F-actin being depolymerized into G-actin in the reverse direction. So at a steady state, concentration of G-actin is the critical concentration. If the actual concentration of G-actin is greater than the critical concentration, G-actin will polymerize into F-actin until the critical concentration is reached. So if the concentration of monomer is greater than the critical concentration, it will drive the reaction, in this case, from the left to right to form the filamentous actin from the monomers. On the other hand, if the G-actin concentration is less than the critical concentration, then the reaction will be driven in the opposite direction. F-actin will become depolymerized to G-actin. Again, until the critical concentration is reached, at which point you will have a steady state. So this is a very simple biochemical reaction, and it's very easy to think about it that way. So what it looks like if you were to measure a mass of filament that are occurring over time. I want to remind you that we are looking at a test tube in vitro. On the left panel, you are seeing there is no seed or nucleus added to the test tube. You have this lag phase, the nucleation phase, where few monomers have found each other and formed a short polymer. Then you get rapid elongation, shown here, and eventually you reach a steady state where the rate of monomer on is equal to the rate of monomer off. On the right graphical representation, now if you happen to add some kind of nucleus to a reaction, and the nucleus could be a small actin polymer or some actin binding proteins that can serve as a nuclei, and we'll talk about these later. But then you can see that you can overcome this lag phase and quite efficiently, and you get more rapid elongation until you reach the steady state. So as it turns out, all of these G-actin monomers are ATP binding proteins. So G-actin monomers can bind ATP, and as these ATP-bound G-actin monomers become polymerized into a filament, eventually the ATP is hydrolyzed to ATP. So the oldest part of the filament will be rich in ADP G-actin, whereas the youngest part of the filament will be rich in ATP G-actin. Eventually, you will get depolymerization and the release of ADP monomers, which can be recharged, and the ADP can be replaced for ATP, and the ATP actin can be incorporated into another element. We are going to talk about the proteins that regulate charging of the actin monomers with ATP in the next part. So one of the questions the cell biologist had for a long time was, what was the role of nucleotide binding capabilities of these monomers? Why should G-actin monomers bind ATP? Did it play any role in polymerization or depolymerization? So a very clever experiment was done in vitro. So G-actin monomers were charged with a non-hydrolyzable analog of ATP. What I mean by this is that it is a molecule that looks like an ATP, but it cannot be hydrolyzed or hydrolyzed to ADP. So it is permanently ATP. Then these ATP analogs, charged G-actin molecules, were allowed to polymerize in a test tube, and then they were allowed to depolymerize. When the cell biologists discovered was that they could still get polymerization of G-actin monomer that harbored the analog of ATP, but they could not no longer get depolymerization. This suggests that the ATP must be hydrolyzed, so the ATP hydrolysis plays an important role in the depolymerization of actin filaments, but not in the polymerization of actin filaments. So, I mentioned that there is unequal growth of the filament at the two ends of the actin. In vivo, the plus end is the fastest growing end, and the minus end is the slowest growing end. In vivo, you usually see is an addition of monomers to the plus end and a loss of monomers on the minus end. This happens for two reasons in vivo. The first reason is that in fact, the critical concentration at the minus end is substantially higher than the critical concentration at the plus end. What is interesting is that the typical G-actin concentration in the cytosol is between these two critical concentrations. So the critical concentration of the minus end is almost always greater than the concentration in the cell. So rather than getting addition at this end, you are going to get loss. Remember that the G-actin concentration is less than the critical concentration if you are going to drive the reaction in the reverse order from F-actin to G-actin. You will normally see a loss at the minus end. On the other hand, the G-actin concentration in the cytosol is greater than of a critical concentration at the plus end. This will drive the reaction towards the filament at the plus end, where the G-actin concentration is higher than the critical concentration. You are going to get formation of filaments until the critical concentration is reached. That is why in vivo, you almost always see addition of monomers at the plus end. Now, this unequal growth results in a very interesting phenomena known as a treadmilling. Now, treadmilling is the apparent movement of monomers through the filament from its plus end to the minus end, just like they were moving on a treadmill. So as you get continuous addition to the plus end right here, you get a continuous loss at the minus end. If you could track some of the monomers, you can easily do this by adding GFP actin. You can imagine that these two green monomers are labeled with GFP, or they are just expressing GFP along with the actin. It will appear as you lose monomers to the minus end and add monomers to the plus end. It will appear as if these two monomers are moving or treadmilling through the filament until they reach the minus end, where they are lost because of the biochemistry at the minus end. So this is known as a treadmilling, and this is a very important process, especially for the cells that want to extend their membranes or when they want to move in certain direction. The actin filament can polymerize in certain parts of the cell in this treadmilling fashion to create forces necessary to extend their membrane in certain directions. So the first reason there is unequal growth at the end in vivo because of the differences in the critical concentration of the minus and the plus strand. So this is what it looks like with the numbers. The kinetic experiment have shown the rate of addition of the ATP G-actin is 12 micromolar per second at the plus end and 1.3 micromolar at the minus end. What it means is that if 1 micromolar of free ATP G-actin is added to the preformed filaments, 12 subunits on average will be added to the plus end every second, whereas only 1.3 subunits will be added to the minus end. So you can see that the addition of ATP G-actin is approximately 10 times higher at the plus end. Similarly, if you just look at this bottom arrow, skinny arrow, which shows the dissociation or depolymerization of the G-actin, at the plus end, the rate of dissociation is 1.4 per second and 0.8 per second at the minus strand. the values are much closer. So you can conclude that the rate of dissociation does not depend upon the concentration of the G-actin. But what is the implication of these association and dissociation rates for actin dynamics? As it turns out, at the plus end, the critical concentration, denoted as Cc plus, is 1.4 subunits per second, 1.4 subunits per second over 12 subunit per second. That is 0.12 micromolar, 0.116 rounded to 0.12 micromolar. So above this concentration, which is 0.12 micromolar, G-actin is added to the plus end and it grows. But what happens at the minus end? As the rate of addition is much slower and the rate of dissociation is closer to the rate of addition, right here, 0.8 to 1.4, the critical concentration, which is the Cc minus, is 0.8 over 1.3, which is approximately 0.6 micromolar. So what does that mean? Thus, at less than 0.6 micromolar concentration, something like in a cellular condition like 0.3 micromolar, the minus end will lose actin subunits. But at the same time, the plus end, 0.3 micromolar, is higher than the critical concentration, and the subunits are going to be added and the chain will grow at the plus end. But it will lose actin subunits from the minus end, and this treadmilling will continue. That means addition of new G-actin at the plus end, and these will move towards the minus end to get depolymerized. So the second reason for the unequal growth rate is that there are certain actin-binding proteins within the cell that add another layer of regulation to the addition of monomers to the plus end and the loss of monomers at the minus end. Now, in this part of the talk, we are going to talk about these actin-binding proteins, so we'll come back to this later. So because this treadmilling, there are some interesting toxins that you can use to assess the role of actin in certain cellular functions. One of these toxins are known as the cytochelacin. It is a metabolite from a fungi. And what is interesting about these small molecule is that it causes depolymerization of actin. So if you take cells, you treat them with cytochelacin, in a very short time, all of the filamentous actin in the cell will become depolymerized. And then what you can do is that you can ask certain questions like, once I depolymerize the F-actin by cytochelacin, will the cells still move? Will the cells still divide? Can vesicles still be trafficked in the cell? As you can answer these questions, you can define the role of actin in the cells. So what's the mode of action for the cytochelacin? What cytochelacin does is that it binds to the plus end of the filament, as shown here, this blue triangle bound to the plus end. This prevents the addition of monomers to the plus end. So it blocks the plus end from growth. What is interesting is that you still have the loss from the minus end and you don't have any growth at the plus end. Eventually, all of these monomers will just come off and you get rapid depolymerization of actin. Another interesting toxin or small molecule that you can use in a similar experiment is known as latriniculin. It is a metabolite from a species of sponge, like a marine sponge, and it is also causes depolymerization of actin. So if you treat the cells with latriniculin, in time you lose all of your filamentous actin. Mode of action is a little bit different than the cytochelacin. What latriniculin does is it binds to actin monomers, so it is not binding to the filament. It is actually binds to the monomers and sequesters them to prevent them from being added to the plus end. So if you add enough amount of latriniculin, you can bind up all the monomers in the cell, so you have no longer get the addition of the plus end because there are no monomers to add to the plus end in the cell. you still get loss from the minus end. So eventually you get depolymerization of the filament. The third toxin that you can use is a small molecule phalloidin. It is a toxin from a fungus, and in particular, it is from Amanita phalloides, or commonly known as the death cap mushroom. This particular mushroom is particularly lethal. You only have to ingest a very, very small amount of this mushroom to become very ill and potentially die. It is actually the phalloidin in the mushroom that is toxic to humans. So how this phalloidin work? So it doesn't depolymerize the actin. What phalloidin does that it stabilizes the actin in the cell. So remember that I told that the cytoskeleton is very dynamic. This is true for actin. Actin filaments are constantly growing and shrinking in the cells, which is necessary for the cells to survive. But what phalloidin does is that it binds to the side of an actin filament. It stabilizes the whole filament, so you no longer have the loss at the minus end. And there is some evidence, but it is not conclusive, that phalloidin also prevents addition at the plus end. So when you add phalloidin to the cells, it is like you are fixing and freezing the filaments in time. So just so you know that the death cap mushroom is very abundant in the Southeast United States, although it's a native of Britain and Ireland. When people eat these mushrooms, the phalloidin toxin initially attack the liver cells. Those who survive, a very few of them usually will require a liver transplant. And the only treatment after ingestion is a lot of IV fluids and a drug called silbinin, which is produced from the milk thistle plant. But the drug is licensed in Europe, but it is still in clinical trials and it is not FDA approved in the United States. So the take-home message is that don't eat wild mushroom unless you are very sure that they are safe. So here is what is the effect of cytochalasin is on the cells. This is a visual representation of the cytochalasin does. So here is a Nomarsky image. Remember the Nomarsky image, or DIC, gives you a pseudo relief image or a pseudo 3D image. In the first panel is the control cell, an untreated cell, and you can see these actin filaments right here. You can see these amazing details right here. Extends toward the edge of the cell like a fan-like distribution. This is a pretty amazing image that you can see really tells you that how these big these filaments are, because you can see them under the light microscopy, like Nomarsky microscopy, without any stain at all. And in the second panel is the same cell after being exposed to cytochalasin, and you can see how the filaments are starting to shrink away from the plasma membrane. And in fact, if you stain the actin in the cell, you can clearly see that the actin filaments are shrinking as a result of the cytochalasin. There is no actin near the periphery of the cell. So the phalloidin is very interesting because you can use it to test the function of actin, because it goes into the cell, it freezes the actin cytoskeleton. Also, the cell biologists use phalloidin as a stain for actin filaments. It is commercially available and you can get them either green, red, or blue fluorescent phalloidin. When you stain the cells, all of the phalloidin will bind to the actin within the cells and you can visualize them with the fluorescence microscopy. So instead of using an antibody to the actin, you can usually use a fluorescent version of phalloidin. And right here is an image in the cells that is stained with a green fluorescent phalloidin, and these large blue circles are the nuclei that has been stained with some kind of nuclear stain like a DAPI or HEXT. So now we are going to talk about the proteins within the cell that interact with the microfilaments. The filaments are interesting in themselves, but it is really the proteins that interact with these filaments and help to regulate the function and mediate the roles of actin in the cell. There are a very large group of actin-binding proteins within the cell that can be placed in the following categories. So there are proteins that regulate the polymerization of actin, and these includes the profilin and the thymosin beta 4. These proteins that can control the length of the actin filaments, including the cofilin and gelsolin. There are proteins that regulate the shape of the filaments, and cause them branch or nucleate into new actin filaments at branching points. An example of this is this ARP2/3 complex. There are proteins that can regulate the cross-linking between the filaments. There are also actin motor proteins, and these are the proteins that use the power of ATP hydrolysis to move along the actin filaments. There are proteins that regulate the stability of the filament, and there are other proteins that regulate the organization of the filament. And in this part, we are going to focus on these first four and have checked them off as the proteins that regulate polymerization, regulate length, regulate branching, and cross-linking. So you all recall that I told you in the first part that these actin monomers can bind a molecule of the ATP. The actin monomers in the filament, in the youngest part of the filament, are going to be bound to the ATP as they reside in the filament. Eventually, the ATP becomes hydrolyzed to ADP, so the actin monomers in the oldest part of the filament are actin ADP. So you can see that fresh new part plus and actin ATP gradually hydrolyzed into ADP to actin ATP. The ATP hydrolysis is important for the depolymerization of the actin filaments because it is thought that the hydrolysis actually slightly changes the shape of the filament so that it becomes destabilized. So one important question that has come up among the researchers for studying the actin cytoskeleton in cells is what is responsible for recharging these monomers with ATP once they come off the minus end as ADP G-actin. Another important observation that has been made by the researcher that study the cytoskeleton is that treadmilling in vivo or within the cell, which happens much more quickly than treadmilling in vitro. So the question is that what regulates or increases the speed of treadmilling within the cells? So there are two important proteins that I want to talk about. It is actually these two proteins that answer these questions. One protein is known as the profilin. It's a protein that is responsible for charging the G-actin monomers with ATP, and then coughlin, its action can increase the speed of the treadmilling. So let's talk about these proteins. So profilin is an interesting protein. It binds to the ADP actin monomers on the opposite side of the protein, opposite from the nucleotide binding cleft. So here you have an ADP bound G-actin monomer. It is bound to the profilin here, which is a small green crescent shaped structure in this figure. When profilin binds to this actin monomer, it actually changes the shape of the G-actin monomer such that the cleft that is holding the nucleotide ADP opens up a little bit and you get diffusion of ADP out of the cleft. Since ATP concentration is fairly high inside the cells, you get ATP diffusing into the cleft when you end up with an ATP-bound G-actin monomer, which can then be incorporated into the growing filament. So what is interesting is that the profilin has a second function because of where profilin binds on G-actin. This function is not shown on this slide. The second function of profilin actually blocks or physically prevents the interaction of this ATP-bound G-actin monomer at the minus end. So profilin has two functions: to charge the monomer with the ATP and to prevent the charged monomer from binding to the minus end. So you remember we talked about in the first part, for all intent and purposes, there is no growth at the minus end because of the critical concentration is so high at the minus end. But another reason is that because profilin prevents the binding of ATP G-actin to the minus end. Okay, so what about this increase in speed of treadmilling? Coughlin is an interesting protein that what it does is that it binds in the portion of the filament where the concentration of the ATP actin is high. So coughlin binds to the older parts of the filament, and what happens is that it binds to these filaments, it changes the shape slightly of these filaments, so they become destabilized and it actually severs the filaments so large pieces come off. So you can think of coughlin as a severing protein. It will sever big chunks of the minus end. What this does is that it opens up the minus end for depolymerization to occur. As a result, you get increased treadmilling in these filaments. So the covalent speeds up the depolymerization by looping off big chunks of filament, exposing more minus ends so that the monomers can come off. So another interesting characteristic of these cells is that the G-actin concentration is extremely high. In fact, in most cells, half of the actin in the cells is monomeric in a G-actin form. If you think of the formula in the first part, there you have been a reaction that is similar to a biochemical reaction where you have a G-actin monomers that are polymerizing into F-actin. You can imagine that if the G-actin concentration is so high, that why isn't all the G-actin found in the filaments? Why aren't you driving the reaction towards the filament? As it turns out that there is a group of proteins that can bind to the G-actin and prevent them from being incorporated into the growing filament. So this controls the formation of the filament in the cell. An example of the G-actin sequestering protein are thymosin beta 4. Thymosin beta 4 can bind to the ATP G-actin, as shown here in this gray structure, is the thymosin beta 4, and prevents incorporation of this particular monomer to the plus end of this filament. But what is interesting is that the thymosin beta 4 bound to G-actin monomer free G-actin monomers in dynamic equilibrium. So if the G-actin concentration in the cell goes down, this will naturally cause the release of G-actin monomers from the thymosin beta 4 and will release more monomer for the incorporated into the plus end. So there are three proteins that regulate the filament formation in the cell. So what are one of the cellular functions that rely on the actin polymerization and actin treadmilling? One of them is motility. We are talking about cells that have the capability of moving. Motile cells such as white blood cells. You can think of motility happening in 4 steps as shown here on this slide. So here is a motile cell. It is bound to the extracellular matrix. The direction of the movement is left to right on this slide, left to right. So in the first step, what you have, what is known as the extension. The extension is characterized by stretching or outgrowth of the leading edge of the cell to form a structure known as the lamellipodium, which is the membrane structure that resembles a foot. So this foot is stepping out, and the second step is the adhesion, where the lamellipodium actually touches the surrounding extracellular matrix and the extracellular matrix component. And then they get out, and then you get the formation of a new adhesion between the cells and the extracellular matrix. You get the forward movement of the cell body. You can think of it as the body of the cell snapping forward as shown as in the translocation. Finally, you have the lagging edge of the cell or the rear of the cell that needs to have deadhesion, where you get the release of the cell from the matrix. So the polymerization of actin and this actin treadmilling is really important in the first step if you have extension of the membrane. So here is another way to look at it. Here is another motile cell. I know it looks like a fried egg. Anyway, in this case, it moving to a direction from the right towards the left, like the arrow. You can see the zone of actin polymerization shown here by all of these pink fibers. As these actin filaments grow, profilin is promoting the growth of these filaments. You have an extension of this membrane, which facilitates the movement of the cell. What's interesting is that the polymerization of actin is happening at this leading edge and not necessarily just by adding the straight filaments, but they are actually branched filaments. So what mediates this branching? There is an important protein complex known as ARP23 complex. It is made up of 7 proteins, and here is a structure generated from the X-ray crystallography of the ARP23 complex. 5 of the protein subunits are shown in the gray, and 2 of them, really important subunit, the workhorse of this protein, is shown in the green and the blue. They are the ARP2 and ARP3. The acronym ARP stands for actin-related protein. So this is how this complex is named, ARP23 complex. It is the ARP23 within this complex that can mediate the branching that is going to generate the forces to drive the leading edge of the cell forward. So here are the steps how this can mediate the branching that is going to generate the forces to drive the leading edge of the cell forward. So here are the steps. That is how ARP2 and 3 works. So you have another protein known as nucleation promoting factor, or NPF. This protein can bind one of the actin monomer. When the two NPF actin complexes bind to the ARP23 complex as shown here, this interaction induces a conformation change of the ARP23 complex, and it attains a high affinity for binding to the side of an actin filament as shown here. Then two NPF actin complexes bind ARP23 complex as shown in here. This interaction induces a conformational change of the ARP23 complex, and it attains a high affinity for binding to the side of the actin filament as shown here, right at the step 3. So if the ARP23 complex is not interacting with NPF actin, then it is in a conformation in which it cannot interact with the actin filament. So now this ARP23 is bound to the side of the actin filament. The actin monomer can then initiate or nucleate actin formation in a different direction. The new branch is at a perfect 70 degrees angle away from the original plus strand. So the new branch that you're generating is a new plus strand right here. And these branches are quite big because you can see them under the EM. So there are two EM renditions of the branched actin. On this slide, you can see that there is a very dramatic micrograph that shows the branching accomplished by the ARP23 complex. So here is what it looks like at the leading edge of the cell. You have an actin filaments growing. You have the ARP23 bound to the side shown here in blue. So you are forming these branches. Here is a profilin in green that is charging the ADP to the G-actin that is coming off the minus end. And profilin is making the ATP G-actin, so you can get further growth at the plus end. The forces generated by the growth of all these branches will push the membrane forward. This is all happening at the leading edge. So let me mention on a couple of other proteins that you can see in this figure here. So you have the cofilin in gray that we talked about, which is the actin-severing protein that promotes depolymerization at the minus end. Another actin-severing protein that works in the exact same way as a cofilin is known as the gelsolin. So you can think that these gray circles as either cofilin or gelsolin. Another protein seen in this diagram is the filamin, and it is a cross-linking protein. So filamin can actually cross-link these branches, stabilizing some of these branches. And then interestingly, you can have some capping proteins right here. And we will elaborate about these capping proteins in the subsequent sections as well. We're not going to talk about that now. But these can cap the plus end of these growing filament and stop their growth. This is really important when the cell wants to change their direction. So let's say you have a filament and the cell is moving in this direction. So it is moving to the northeast, and let's say the cell receives a signal that it needs to change direction and move more towards the north, straight north, or more towards the northwest. What will happen is the cell can recruit capping proteins to stop the polymerization of these filaments. So now you have a shear force going northwest direction, and the cell will change its direction, or the plasma membrane, instead of getting pushed this way, it will now be pushed on towards this way. And it may do in response to a particular chemoattractant, for example. What is really interesting is that some intracellular pathogen that can hijack this actin polymerization system to move throughout the cell. And one of the example of one of those pathogen is the Listeria monocytogenes. The Listeria monocytogenes is a bacterium. It is a foodborne pathogen, and you often hear of recall of dairy products or recalls of processed meat like deli meats that might be contaminated with the Listeria. So Listeria is taken up by white blood cells, the immune cells such as macrophages, by phagocytosis, and they end up in the phagosome. You'll remember from our previous discussion of endomembrane system that the normal route is that the particle in the phagosome are trafficked to the lysosome and degraded. But Listeria has a very interesting protein on their cell surface which is known as listeriolysine. Once the Listeria bacterium is inside the cell in the phagosome, that protein can actually lyse the membrane of the phagosome and the bacteria can escape into the cytosol. And in doing so, it never gets trafficked to the lysosome and it never gets degraded by the hydrolases in the lysosome. Then what can happen, we will look at this molecular level in a minute, the bacteria can induce actin polymerization and treadmilling at one end, and it will propel the bacterium through the cytoplasm to the plasma membrane. The force is so strong and Listeria is so good at activating this host cell actin polymerization machinery so that it can actually get distortion of the plasma membrane, and the neighboring cell can take this up by phagocytosis, and then Listeria can escape again into the cytosol and continue on. In doing so, Listeria can move from cell to cell within the host, practically evade the immune system because it is never exposed to any antibodies. This is what it looks like on a fluorescent micrograph. The small red circle that you can see is the Listeria bacterium, and behind them are these green actin tails. Sometimes these actin tails are also known as actin comets because they resemble the comets. The bacterium itself is generating the treadmilling of actin behind them to propellate. Just see the supplementary videos and you will have a very clear understanding that how amazing these actin tails are in moving these Listeria monocytogenes. So how is this happening at the molecular level? As it turns out that the Listeria have on their cell surface another protein known as ACT-A. This is shown as the pink circle here, and ACT-A is a very potent activator of the ARP2/3 complex. Remember, a few slides back I told you that ARP2/3 complex had to bind to the nucleation promoting factor NPF for its conformation change, and once the conformation is changed in a way that it can bind to the side of the actin filaments. Well, as it turns out, ACT-A will activate the ARP2/3 complex, and in doing so, a lot of ARP2/3 binding to actin filaments in the region around the bacterium in the cell will happen. So there is a rapid growth of these branches, and as these branches are growing at the plus end and depolymerizing at the minus end, the bacteria is actually being propelled forward on this actin tail. So this shows the whole of the bacteria, and this is the actin tail behind it. So actin polymerization is really important for the motility. It is also really important for the maintenance of the cell shape and the changes that occur in cell shape. Here are scanning EM of the platelets. I think if I had just shown you these three panels and not told you that they were platelets, you might think that they were different cells. But in fact, these are all platelets. In the first panel, on the left side is an unactivated platelet. But once the platelet is activated as a result of a vascular injury, let's say a cut, they will bind to the site of the injury and they will form this structure to prevent the injury. Eventually, they will spread out to cover the vascular injury. A lot of this is mediated by the changes in actin. In particular, it is mediated by a transmembrane protein complex known as GP1B9, which can interact with a clot on the side with actin on the cytosolic side and the platelet membrane. It connects with the actin through filament. So this is another role of filament, which is a cross-linking actin binding protein. So how does the cell coordinate all of these different functions of actin? What can regulate actin polymerization? As it turns out, there are signals from the soluble factors outside the cell, signal from other cells, and a signal from components of extracellular matrix. All of these signals act through the signal transduction pathway to regulate the actin polymerization in the cell and recruit actin cytoskeleton for motility and for changes in the cell shape. So these signals are usually received at the cell surface by the plasma membrane receptors and propagate within the cell through signal transduction pathways and communicate the signal to the cytoskeleton. Often more than one receptor conveys these signals and depends on which receptors are communicating. You might get changes in the cell shape or changes in the movement of the cell or even contraction of the cell as we just saw in those platelets. Or you might even get reorganization of filaments that might affect the position of organelles or cause organelles to move in the cell that regulate the movement of vesicles. So when we talk about cell signaling, later in the course, we'll talk about the details of the cell signaling pathways. So we are going to be looking at specific receptors and what downstream molecules regulate the signal to relay from these receptors. This concludes Module 4, Part 1.